here on your, I don't know, internship, RU, whatever you want to call it. And what we're going to look at today is an interesting topic. It's a topic that often people want me to present because of its interest. It's on cancer and cancer drugs, nanotechnology drugs in cancer. And as we could all probably envision that we could spend a lifetime studying cancer and then never have a cure, because certainly a lot of people are doing that today. But this is a nice overview to take a look at things. And again, we're limited in time, but we can cover some neat things in here, and I think they're really good lessons for you. Not only here in the academic world, but it's a tragedy that, just life itself, deals people the cancer card. So you might have cancer yourself or members of your family or friends. So it's nice to know about these things and see how they work. So this knowledge is nice to have around. So that's what we're going to talk about today. So specifically, we're going to look at nanomaterials for bioapplications. And on my front slide here, we're going to look at that. That's actually a nanotechnology unit right there. So that's our nano drug, and that's called a liposome. And what a liposome is, it's an organic nanomaterial, organic nanoscaled material, and it's kind of like a jelly donut, but it's a jelly donut that has poison in it. And our goal is actually to take the jelly donut and put it into the cancer. The cancer eats the jelly donut, and then the poison's in there. So basically, this is like a trap for the cancer drug to go into the cancer, and it kills it. So it's kind of like baiting rats or something like that with poison. So I wanted to remind you, when we're looking at this is the end of your time here for your stay here, looking at the technology, having an overview here in the summer. What we try to do with materials is we look at the properties, the process, and the structure to be able to make our particular application. So in this is a generic drawing of what we would like to do, but specifically what we would like to look at, as far as properties, we're looking at the hardness and the elasticity and the biocompatibility of our drug, and we're designing that into our structure. We also, if we look at example characteristics of structure, we're going to look at the size and the size distribution, the shape, and certainly, I guess, the number of defects, or the numbers that are out of size of the Gaussian distribution of size of the particular drug. And then we're going to look at the material itself. And all these are important to us, like the rate of cooling, the annealing, and the extrusion. And again, this is a generic chart that represents, just some brainstorming of what it takes to make nanomaterials. And those ones, specifically those items we're going to discuss today, to see how we would actually make this particular organic nano-sized drug for cancer. So what we're going to take a look at today in the first section, we're going to take a quick look at biocompatibility. So I assume there's people in the audience here watching this video that don't have a really good grasp of biotechnology or cancer or cellular function, because they might be brilliant people, but they're electrical engineers and they never get that opportunity to take classes in biology. So for bioengineers, this will be a little bit boring and probably, certainly not on their scale. And they can see that I'm going to abbreviate a lot of stuff for time's sake. But we're going to take a quick overview of like what cellular interaction is, and we're going to look at the size of the cell, a generic animal cell. What's the importance of DNA, and what is cancer. So boy, that's a lot. That's probably a lot to cover in a week in a biology class, those particular topics. And I'm going to cover it today in 20 or 30 minutes. So hopefully we can grasp the important details and get them out there, because that's a big chunk of technology we're going to try to assimilate in a rather quick time. And then I'm doing that to set up the second part of my presentation, which is we're going to try to look at this material called a liposome. And when we do manufacture, so this is a manufacture, or at least my discipline is in manufacturing on the nanoscale, we want to look, how would we manufacture in the nanoscale a liposome? And that's different from a lot of the things that you've looked at so far in your stay here in the summer, because we looked at things like plasmas or vacuum technology or lithography, dry etching, those kind of topics. And this is divorce of all those things. So this is a different topic, But again, when we take a look at, you know, nanotechnology and material science, it overlaps in these areas. So it is another separate, maybe a separate subset from the other things that we discussed here. So I think it rounds out your orientation and gives you a well-rounded look at, you know, nanomaterials. So let's get a start in on that. So first of all, we want to define what biocompatibility is. And biocompatibility is the ability of a material to perform with the appropriate host response in a specific application. So to do this, to engineer this biocompatibility into the material, the nanotechnologist must amalgamate the understanding of the materials in a biological response. So we take foreign objects like gold nanoparticles and we put them in the body to do a specific job for us. And then we have to say, well, the body probably doesn't want to have gold nanoparticles in there. How would we hide them, or how do we coat them, or how do we deliver them in the body? Because the body might, per se, reject those. So we look at the body's response. So sometimes we do want the body to reject things. And for example, if we have a screw and put it into a bone, like you break your bone, you put a screw in there and it's bioengineered screw, specialty screw to hold your bone together. But what the body will see that and kind of like reject that and form bone cells around it, kind of like a scab in some senses, so that it protects the body from that. But what it's going to do is heal that screw into your body. So that's one way of looking at it, that the biocompatibility in that issue, we have a dramatic biological response to coat over that object. We could look at another version of that, which is still biocompatibility, and if we put a catheter or something in your heart or in your arteries or whatever, we don't want the body to heal over that and to clog it and to grow things over that. We would have that as the exact opposite of that. So biocompatibility could be really rejection of that or acceptance of that body part, or this foreign object in there. So biocompatibility is both, you know, a positive and negative thing, how the body would react to that. So you can have like ignore or dramatic reaction. And that's what we have to do as nanotechnologists. In our materials, we got to program our materials so when we put them into the body, we're going to have the reaction that we want. So that's really critical to understand that. So the cell size itself, when we look at the cell in the body, the cell itself is usually about 10 microns or so. That's a good guess, but there's all kind of different cells in the body. But basically the cells in an elephant, or cells in a mouse, or a cell in you, they're all about the same size. So like, for example, a red blood cell is about 7.5 microns in diameter. And what's nice about that is if we made like some kind of lab on a chip, or some type of discovery for mice or for horses or for cattle, we could probably directly apply that to people because the cells are at the same size. And why, a good question is, why would the cell size be moderately standardized throughout the animal kingdom? And that's because it's efficient. And so we need to have this cell size that carries out the biological response of the body. So what we have tried, what nature has done, is actually gave us a specific size that's really good. So here's a nice relative particle size, and I have this slide in a lot of my drawings because I try to interrelate things throughout the program that I teach here at Penn State. And it's really important to know these cell sizes. So for example, human hair is about 100 microns or so, and a blood cell's about 7.5 microns. When we start looking at bacteria, bacteria could be the same size as a standard cell, or maybe 20% of a standard cell, or 100 times smaller. So 0.2 to 10 microns for bacteria. But when we look at viruses, they're probably 2 to 100 nanometers. That's really important, and I'd like to talk about viruses here for a second. So we all know that people, and you say, I'm sick, I have a virus. So that's a common theme. And viruses are very difficult to cure, right? So we have antibacterial things, but viruses, usually you get a virus and the doctor just says, well, take it easy for a while and your body will respond and hopefully be able to react to the virus and kill it on its own, because it's difficult to get antiviral drugs, right? So those things, and the reason why they're so dangerous is they're on the scale Viruses are on the scale that they can kind of enter cells. So let me say that again. Viruses are of the size scale that they can enter cells. So they kind of pose a danger because they kind of have free reign. So they have a certain amount of autonomy to go throughout the body into the cells and then do their things. So, and usually negative things, the viruses will give you some kind of illness. And that's really bad. But what we can do is we could use that idea and say, if we want to make a drug, let's make it at the same size, because now we know that that drug could probably be implemented into a cell. So as far as scaling, what I'd like to present to you, this idea right now, that a cell is approximately 10 microns. And again, that's just a rough number. Does everybody see that? So I'm just throwing out a number for our mental exercise. So saying that a cell is around 10 microns, when we look at if we would make a drug that was as big as a virus, we should be able to implement that inside a cell. And that's what we're going to do today. So a key to today is the realization that the drug that we're going to make a man-made drug that's as big as a virus. Does everybody see that? And that's our goal. So let's discuss that goal a second. That's crazy. Does everybody see that? Like, we're sitting around, drinking coffee here on a Thursday morning saying that, what we want to do for a living, that we're so cool as like engineers and scientists that we can make drugs as big as a virus, program those drugs so that we can specifically send them to an area in the body to do some healing thing. And then the one I'm proposing to you today is to do cancer. So this is a pretty wild story. Does everybody see today's presentation? We want to be like the craziest of all ninja, engineers and scientists, that we could create drugs as big as a virus and then be able to send these drugs that are as big as a virus into a patient that has cancer and then to kill that cancer. And again, that's crazy, but it's not a pipe dream. I'm going to show you what we do, and it's us. We take responsibility to be able to do things like this. So this is why you get educated, because these things are possible today, and we need people like you, the youth, to be able to carry this forward and to be able to continually make these drugs, because these drugs are cool. Does everybody see that? So we're going to talk about drugs that they're using today in hospitals, not in research. Today at the local hospital up the street from me, they're probably administering these same drugs to help cure cancer. So this is where the bar is. Does everybody see that? It is, again, kind of wild that we could sit here and say that we're going to study so hard to put our efforts in that we can actually cure cancer. You guys, in 2 or 3 years, you could be making this stuff and impacting society really greatly. So I think you should be proud of your work here in the summer and proud of your work as an engineer, because you can do these things. Does everybody see that? So I think this is a really cool presentation, so I'm glad you're attending today. So this is a generic animal cell off of like an NIH or NSF website. And what this basically says, inside the cell, we have things like the nucleus. And in the nucleus, what we're going to do, that houses the DNA. And the DNA, we could say, is like an analogy. That's like your hard drive. And then what the DNA is going to do is that hard drive is going to express like a program. And that program is like that subroutine is going to be RNA. And RNA is going to be like a program, like Microsoft Word package or something, or like Outlook, or some kind of email thing, or like Google Mail. And what that's going to do, the RNA, that program that we're going to call off the hard drive, what it's actually going to do, its thing is not to get email. What it's going to do is that RNA is actually going to make proteins. And those proteins are what's expressed in your body. And the proteins, if you would talk to a biologist, they would say that proteins are life's currency, and proteins make things happen, and that's communication. So I think that you should know that proteins are things like hormones. So the definition, a hormone is a protein, and that would allow your body to do specific things. So the body uses, like sugars and stuff like that to do the energy as a driving thing. But basically, if you would say to a biologist, and again, I'm only given 90 minutes or so to give a presentation on biology, it's very difficult to do, but the thing that I'd like to have in your mind is, proteins are life's currency. And what they do is they lock and unlock things and make things happen. And they're like a cascade agent that allows your body to do things. Like, the proteins, if you get a cut in your hand, the proteins are going to do this signaling, etc. And the proteins are going to clot your wound. And the proteins are going to send signals to the cells to like regenerate that wound, etc. So the proteins are like the movers and doers in your body. And that's especially true to know. Does that make sense? The other thing that we want to know, and I'm going to give you in the presentation, I want to show you multiple times so that you get these big ideas. What your cells have to do every once in a while is they die. So what they do before they die, they probably make a sister cell so that replaces your cell. So every so often, all your, like, not all, but periodically, things like some of the cells in your liver die. And they die because they get old and they need to be replaced. So what they'll do is they divide, and then maybe the old one will die later. It gets defective and it's killed off. And then that's called apoptosis. So your body detects that a cell isn't doing the right job or some other thing like that. And what it'll do is self-kill itself. And that's important, because if your cells get old and start doing funky, goofy things, then you don't want that to happen, because you're going to start mutating. Does everybody see that? And that might sound funny, but the reason I'm talking about this funny story is that is what cancer is. Cancer is rogue cells that don't know to kill themselves, that rapidly multiply. Does everybody hear that? So it's kind of like a funny story, but the story is, what is cancer? Cancer is a mutation that doesn't follow the standard rules of cellular division, doesn't do the standard rules of cellular life, because there's a finite life in a body of a cell. And what cancers are is they don't have the ability to kill themselves, and they don't have the ability to turn off division. So what they do is they grow these tumors. And those tumors basically and literally suck the life out of you. So it's kind of like one of these movies, I don't know, what's the movie, like Aliens, where things grow in you and something pops out your chest? But it's not going to be that crazy. But you get this tumor in there, and it's like an alien form that's in your body. Does everybody see that? And it's going to take over and kill you. And that's what we don't want to have. Does everybody see that? So the drug that I'm proposing today, and of interest in this presentation, our drug, what it's going to do is, during that, when the cancer goes to divide its cells, it's going to prevent the cells from dividing. Therefore, the tumor is not going to grow. Does that make sense to everybody? So, first of all, what I have to do is define what is cancer. So cancer is unregulated cellular growth that you don't want. And what can I do to cure cancer? Well, there's multiple things that I can do to cure cancer. But one of, like, say, 7 different themes that you would have in cancer is, what you can do is keep the cells from dividing. Does everybody see that? That's one way of killing cancer. Another way is hit it with radiation and just kill it. But that's difficult, because the radiation also kills healthy cells around it and has negative impact on the patient. What we're trying to do is create a drug as big as a virus, put it into the cancer, kill the cancer, but not harm the patient a lot. That's like a $1 billion idea. That's like so noble of an idea, it's ridiculous. We want to kill cancer, but not harm the patient. So this is an important presentation, and I'm proud to present it. So there's different cells in the body, and we're not going to worry that much about it, but they do different things, and they all have their own same function, but they have a commonality that they often divide and stuff like that, but they do different things in the body, and we don't have time to discuss it, but I just wanted to make you aware of that. So these are the typically different cells that are in the body. We're not going to key on that. We're just going to say a generic cell. Does everybody see that? But there are different sizes, and they do different functions. Probably one of the ones that we want to think about is, though, and why I have this slide up here, there's a thing called the epithelial cells and endothelial cells. And what they do is they line the body cavities. So like in your bloodstream and all that other stuff, when they form like scales like on a snake or like shingles on a roof. Does everybody see that? And they have to be tight. Like you have to have your shingles on your roof real tight. And if a shingle blows off or if there's a hole in your roof, water leaks in. Does everybody see that? So endothelial cells and epithelial cells, what they do is they're gonna be like that sealing thing. Does everybody see that? That's going to be important in my presentation. So there's a lot of key ideas in my presentation that might seem odd when I'm introducing them. And I'm talking about this sealing thing that looks like shingles on a house and scales on a snake on the outside. But these are going to be on the inside of you. And that's how your body is formed. And that's going to be important to when we discuss how we send this drug in. Because actually what we're going to do is, in cancer areas, cancer grows so rapidly that those overlapping cells don't overlap. And they're going to have like holes in them. Does everybody see that? So when we want to kill cancer, what we want to do when we look at cancer, just as an overall theory, like in engineering or in bioscience, we want to say, how is cancer different than healthy tissue? And when I identify these differences, what I can do is take advantage of these differences and then make a drug that will target specifically these differences. Does that make sense? So the difference I'm trying to identify for you right now is we don't have, like in the matrix of the network, like cancer or surrounding medium, like muscle cells, they're not the same as healthy muscle cells. The cancer area is defective and has holes in it. We're going to send our drug in those holes. And since the healthy tissue doesn't have those holes, this poison won't go into the healthy tissue. Does everybody understand that? So we're trying to identify why is cancer different. And when we identify that, we're going to use those differences to our advantage, and we're going to send in the drug in those specific areas. So this is a membrane structure, and we're also going to say this is the basis of the drug I'm going to make. So this is a cutaway version of what a cell membrane looks like. So this is a cell in your body, like a blood cell or something, and the blood cell is like a water balloon, right? And then the balloon itself, that outer coating, this is what it looks like. And interestingly, that outer coating and what every cell is made out of, it's made out of these things called a lipid bilayer. And what a lipid bilayer is, it's actually a part of nanotechnology, certainly, and it's a subunit, and it has a phosphate head and fatty acid tails. And what these tails do, like oil doesn't mix with water. If we would put this in a solution, these lipids, when we put them in a solution, all the fatty tails are going to go to minimize their energy, and the phosphates are going to point to the water. So basically, cell membranes follow this nanotechnology rule set by the lipid. Does that make sense to everybody? So the lipids have two different ends. They have a phosphate head that likes water, and they have fatty tails that don't like water. And with looking at energy or thermodynamics in energy, this material, when you place it in water, it's going to form a thermodynamically, energetically favorable unit. And what that's going to do is point the phosphate heads to the water and the tails into themselves. So this is how cells work. So this is a membrane structure, and nature does this, and it's been doing it for billions of years, right? This is like in amoebas. This is how things are formed, right? So what we do as engineers, we look at nature, and we say, hey, let's look at that. And now my second statement here is this a good drug design? Yeah, it's a good drug design. So we're using the basis of how cells are formed. We're going to copy that, and that's going to be the basis of my drug. So let's go back up to my first slide. So this is the cell membrane. Does everybody see that? Now let's look at my first slide in my introduction. This is the lipid bilayer, and this is my drug. So my drug is a copy, or it uses the same rules as a cell. So how biocompatible is this? It's like, it's the definition of biocompatibility. It's the definition, because what I'm doing is copying the structure of the cell and using that as a drug. And now let's take a look at this other slide. My drug is as big as a virus, but it looks like a cell, and I'm going to be able to put it into a cell due to the scaling. Does that make sense to everybody? So my drug is going to be as big as a virus, and I'm going to make that, and it's going to look like it's a piece of jelly donut that the cell wants to eat, because I made it out of something that's not so foreign. And not so foreign by definition is a lipid, because that's the cell membrane. So I'm doing a lot of crafty things here. Does everybody see that? There's a lot of, in some senses, built-in wisdom in this ability to make a liposome, because I'm doing exactly everything we need to do. And you know, what I'm trying to do here is beat cancer. And certainly that's a noble thing to have as a goal. Does everybody see? So this is the phospholipid structure. And if we look at the head over here, the head is a phosphate, and the head likes to go to water. And then the tails are made out of fat, so they're going to point in. Does that make sense to everybody? So that's what we're trying to do. We're trying to make a drug that's biocompatible and is energetically favorable, because we don't have that much energy in the body. Does that make sense? And we want to use like the rules of water and self-assembly. So this is the animal cell right here. And again, we went over this, and what we're importantly looking at is we want to like tie up the DNA when it goes to do cellular division. So I'll present some slides on that when we get to it. So the DNA itself consists of a number of nucleotides, and what these nucleotides do is they actually encode for protein synthesis. So for every 3 nucleotides, those things are like a digital code, like in computer science. Like if we have, you know, 1001 or something like that, you know, 1001, that would be 9, right? So 8 and a 1. So that would be the code for 9 in a binary system, right? So what we do is we use this, and we're going to use these base pairs in here, and every 3 base pairs are actually going to be encoded into an amino acid. And the amino acids linked together are going to give us a polypeptide. And when a polypeptide is in a water, it's going to move and then fold into a 3-dimensional structure, which is then a protein. And these proteins, like I discussed before, is going to give us the possibility to do communication within a biological system. And so this is a self-assembly rule. When we break our DNA is a 2-tiered system. So we have a complementary strand on either side. And by complementary, I'm going to define that as one area has like this guanine. It has a role that it's only going to form its sister on the other side is going to be the cysteine. So a G forms to a C, and a T forms to an A. And if I have an A on this side, it's going to be a T on here. So it's kind of like a reverse image. Does everybody see that? So there's a role of G's and C's and a roles of A's and T's. So if you knew one side of the DNA, one side of the strand, you know the other strand. And importantly, and what nature does is when a cell divides, it takes those strands and unzips them, and then it makes new DNA because it would know what the other side looks like. Then what you have is, in some senses, I guess a loose word would be a cloning. So what we have to do when we do cellular division is we have to clone the DNA, because each cell needs the DNA. So you'll have the original cell and then the daughter cell. Does that make sense to everybody? And again, when we look at cancer, cancer is unregulated cellular growth. Does everybody see that? But you need to divide the DNA to have cellular growth. Does everybody understand that story? So for a cell to divide, you need to divide the DNA. So for a cell to divide, you need to divide the DNA. Our drug is going to actually be like a tie wrap, and it's going to prevent the DNA from dividing. And if we tie up the DNA and don't let it split, then the cells can't split. If the cells can't split, you don't have unregulated cellular growth, you cured cancer. So a lot of people just in the general public, they always discuss cancer and say things about cancer or whatever. They might not know that simple definition. A definition of cancer is unregulated cellular growth. So a question is, how do we cure cancer? Well, we inhibit cellular growth, and that cures cancer, or one way of curing cancer. And now my question to you is, or the lesson here is, how do I cure cancer? Well, what I can do with the DNA is I can prevent the DNA from splitting, and if it doesn't split, then we don't get two cells, so therefore cancer is cured. So what my drug is going to do is prevent DNA from splitting. So that's what I'm after. And why do we want to, why do we care about DNA? Well, certainly we care about it because it controls, well, it doesn't control cellular division, actually proteins do. But the proteins themselves, when the DNA is made, it makes RNA, and then the RNA makes proteins. And then protein is the currency of life. So that's the central dogma of biology. The central dogma of biology, like for biologists, they're like Ohm's law. Like the electrical engineer, we got like a holy law, right? Of like Ohm's law. That's something that, electrical engineers really dig on. They like Ohm's law and stuff. But for biologists, their central dogma of biology, the central dogma of biology is DNA to RNA and RNA to proteins, because that's just foundational in biology. Does that make sense? And that's what we want to do, is we want to disrupt a lot of these cycles. And there's other ways that we can disrupt these cycles in cancer, but we're specifically going to talk about tying up the DNA so there's no cellular division, and they would call that intercalation of the DNA. And this is a codon table, so like for every 3 data bits, like those G's and C's, you can see this table right here, and like the T is replaced by U in a conversion, so you don't have to worry about that much. But basically, when we look at these encodings, they're going to give us the different amino acids. And then a bunch of amino acids together give us a polypeptide, and a polypeptide is going to give us a protein. So that is showed here. So what we're going to get from the DNA, we get an RNA segment, and the RNA is going to encode for amino acid assembly. And amino acid assembly is going to give us polypeptides, and they're going to give us the proteins. And proteins are this, the essence of life. So this is why these things are important. So the membrane structure, when we do communications, things go in and out of the cell generally through a term called endocytosis. And what we often do is a protein will come over to the cell, and these cells in this area right here, they might have something that looks like a garage door or a door on your house. And what the protein does is it unlocks the door, and it'll allow transport between the cells. So to carry out life, the cells have to communicate with each other. What do they communicate? They're communicating like make a hormone, send out a hormone. I have a cut, send me some blotting, material. I have a damage in my knee, heal my knee, or I have a cut, heal the skin. So these proteins sense these things, and they allow your body to interactively carry out life functions. So that's why these things, and on the surface of a membrane, these communication things are important. So the point that I would like to get across is what we have to do is we send on our drug, and our drug has to have the ability to go into the cell. So it has to be the right size. So cells readily accept things under 100 nanometers. When they're like 500 nanometers, they have a difficult time going into a cell. What we would like to do is we would like to take our drug that's under 100 nanometers, send it through the cancer cell membrane. When it goes into the cancer cell, inside the cancer would like to take that subunit in that looks like a jelly donut, and it would like to take the jelly donut apart for energy. And it wants to reuse the lipids, it wants to reuse the protein in there, it wants to use the sugar in that to do its work. Does everybody see that? So loosely, my story by analogy is, I'm going to make a drug that looks like a jelly donut. And it's going to look so good that the cell is going to say, hey, and it's going to have maybe communication on it, maybe. And what it's going to do is allow it to go into the cell. And once it does, the cancer cell is going to say, oh, good, here's a jelly donut. And it's going to open up the jelly donut, and it's going to release this stuff that's going to tie up the DNA and kill the cell. Does everybody see that? That's the big picture. So that's why these cell membranes are important to us. So this is the DNA replication. Again, this is before cell division. The DNA is together in the double helix that we're all familiar with. Does that make sense? But when a protein sends a signal over and says, hey, I want to divide, and cancer does that all the time. Cancer has unregulated cell division. So it's always saying divide, divide, divide. So this will have this division right here. What my drug is going to do, where this area is, where we see this pink arrow in the second frame, what we're going to do is it's like a tie wrap or a chain or handcuffs. It's going to tie the DNA up so that it can't split like this. Does everybody see? And if it can't split, then we don't have protein synthesis. I mean, cellular division. Therefore, we don't have cancer. So this is cancer. So as a definition here, cancer is known medically as a malignant neoplasm. It's a broad group of various diseases, all involving unregulated cellular growth. In cancer, cells divide and grow uncontrollably, forming malignant tumors and invade nearby parts of the body. There are over 200 different types of cancers known to affect humans. And that's important, and the term metastasis or malignant is important. And basically, when a cancer grows unregulated, that's an issue, and you're going to get a tumor. But actually what kills you is when that tumor moves to another part of your body. So for example, you could have like tongue cancer or lip cancer, and you get that from chewing tobacco or cigars or something like that. And that's an issue because you have cancer in your mouth. But probably what's going to kill you is when that cancer moves into your liver or moves into your pancreas or moves into your brain. And those are when the tumor moves from, because it doesn't know to stay at one place, it'll move into some other area, and then its unregulated growth will kill you. So that's why if you have like tongue cancer from chewing tobacco, so here's a public service announcement: don't chew tobacco, don't smoke cigarettes. Does everybody hear that? Because when you do, you get like a cancer in your lip or on your tongue. But that's going to break free and move to your brain or your liver. And when it gets to that area, because it's stupid, the cancer, it's going to grow, and that tumor is going to kill you. Does everybody see that? So that's what metastasis is. So cancers get really, really serious when they move. And then when they move and have unregulated cellular growth, like I said, it's like a foreign thing, like in that movie Aliens. It's going to kill you. Does everybody see that? And that's what's bad. So cancer expression themselves, there's 22 tumor types. They have different multiple metabolic expressions. So basically they have like protein signatures that they express. So when you have cancer, often what they do is they measure your blood and they look for a particular protein expression. And if they say there's a high amount of a certain protein in your body, then they can extrapolate that you have a certain cancer. Importantly to my discussion is a tenet of cancer called the enhanced permeability and retention. And at this part, what happens is the cancer cells themselves, what they do is they grow, and when they take in stuff, they don't have the ability to get rid of waste byproducts. That's important because that's one of the reasons that nobody is able, that cancer grows and it's hard to cure, is because it has all this junk and waste material in there, and it's at a positive pressure. It actually mutates often. Does that make sense? So anyhow, let me move on to the next slide. So this is the cellular division. So when we go through cellular division, when a cell divides, we have to go through this process. And what we're going to do is, when we do this DNA synthesis thing to like split the DNA, what our drug is going to do is cut that down. Does that make sense? We're not going to allow that to happen. Therefore, we don't get unregulated cellular division. Does that make sense? So here is the, if you look at cancer in general, here is the what happens in a cancer scenario. So these are the different types of drugs and the different areas in a cell cycle. And as like a medical person or a nanotechnologist, what we do is we would go in and we look at these areas, and we go in and we go ahead and we identify these areas, and we make a drug that stops one of these points. Does that make sense? Like before I said, there's a handful of areas that we could specifically make a drug. So here's a drug like cyclin D. And what it's going to do is interrupt that cycle at this point, at the G1, G0. And that's what we're going to do. Does everybody see that? So when you talk to doctors, or I hope you never have this happen, but somebody in your family or you have cancer, they would identify these families of drugs, because these are the families of drugs that are going to be able to do the cure for us. Does that make sense? And so now this is the second part of my presentation. And what I'd like to do right now, just because, in the presentation, I'd like to take a break right now. Does that make sense? So we're going to take a short break to give everybody a bathroom thing and to get a cup of coffee. And then I'm going to come back and I'm going to talk specifically about the liposome and how we make that. Is everybody good on that?